Okay, with this video I want to explain to you what a capsule is, why it would be beneficial to bacteria to make one, and then a little bit about what we can do to the cells so that we're able to actually visualize their capsule. Okay, so a little shorter pre-lab for this lab. A glycocalyx is a general name, a general term given for a slimy substance secreted outside of the cell. Okay, and it's extremely common for bacteria cells to make some kind of glycocalyx because it does give them some advantages. So lots and lots of bacteria make some form of glycocalyx, whether it's considered a true capsule or a slime layer or somewhere in between. So this is really a continuum between the two. Like a true capsule, you're going to have this great big, puffy capsule around the outside of the cell. And maybe a cell with a slime layer, would just be really, really tiny. So there's lots of in between there. A true capsule is just a little bit more rigid, a little bit more tightly bound, larger perhaps, and a slime layer is a little bit more loosely organized. Okay. Regardless of whether it's a true capsule or a slime layer, what's the composition of a glycocalyx going to be? Most of the time, it's going to be made up of polysaccharides. Hard word to spell. Okay, so a sugar composition. Now there are some exceptions to that. There are a few genera that make capsules that have a lot of proteins in them, but most often they're sugars, polysaccharides. And most of the time they are non-ionic, meaning they have no charge. So that makes it difficult for us to see them just by attaching some kind of stain to them. Okay, so instead we try to stain the background so that the capsule stays unstained. Three major functions of the capsule, and of course the slime layer is going to do all these things to a little bit less extent. One big one is adherence, maybe to some part of the host. It could be, say, a bacteria that tries to colonize our mouths. We have hundreds of bacteria that do that. If they make a nice sticky capsule, they're going to be able to adhere to our teeth, okay, instead of getting washed down into our digestive system. Okay, so adherence to host, perhaps adherence to other cells and help it form a biofilm as well. All right, it makes the cell less vulnerable to phagocytosis by white blood cells. I think your book calls the white blood cells PMNs. That stands for polymorphonuclear leukocytes, which is kind of another name for neutrophils. Okay, let's just call that a white blood cell. All right, so any of our non-specific, host defenses, white blood cells are going to be less likely to phagocytize a bacteria that's covered by a capsule. It just has a harder time getting a hold of it. A third function of a glycocalyx, it's going to make the organism more resistant to drying out, desiccation. Okay, think about an organism that's coughed out of our bodies into the environment. If it has a capsule around it, it's going to stay moist for a little bit longer, and it's going to be more likely to survive before it can enter another host. So those are three big functions of the capsule in bacteria. In your lab, on page 23, there's a question in the center of the page that asks you to describe the colonies of the organisms that you see. And what that question is getting at is that typically organisms that produce a big, thick capsule are going to look kind of shiny, slimy. Okay, we use the term mucoid to describe these colonies. Okay, so a lot of times students read that and they think, describe what the cells look like. Remember, when I ask you to describe the cells, that's different than describing the colonies, what we see with the naked eye. So you can't always tell by looking at colonies that the organism's a capsule producer, but that's just a hint. Sometimes, when you touch these colonies with your loupe, it kind of looks a little bit slimy, and then it's pretty obvious. I don't know what organism this was, but it may not be the one that we're using in class. Okay, so today you are going to try to capsule stain 2 different organisms. We're going to look at them under that oil immersion. We are going to do one slide that we're going to mix in some Klebsiella oxytoca. Okay, Klebsiella is a genera of organisms that usually make a nice thick capsule. But we're going to compare that on a second slide with our Staphylococcus epidermidis. Okay, that's typically not considered a capsule producer. You may see a little bit of a slime layer, but hopefully you notice a difference between these 2 organisms. So you're probably going to want to label one of your slides, KO, and label the other one SE or something like that. Okay, so remember, the reason that we have a whole new capsule staining procedure is because when we gram stain, we can't visualize the capsules. And the reason is when you're gram staining your cells, remember, we always heat fix them. Well, when we prepare our cells for the capsule stain, we are not going to heat fix because that heat is going to destroy the capsules or possibly distort it beyond recognition. So that's one big change that we are going to make. When we prepare our cells, we're actually going to put a drop of Congo red. Remember, that's an acidic dye on one end of your slide. And then with your loupe, you're going, and really doesn't matter if you use your loupe or your needle. Our cells should be on a plate today. But you're going to mix some cells into that drop of Congo red. Go ahead and spread that out, again, like nickel size. Better you spread it out, then it'll take a little bit less time to dry. And then we're going to do, remember, like I said, we're not going to heat fix it. So no heat fixation. Okay, and then we're going to do what's called the push slide method, where you're going to take a second slide. I don't know if I can draw this very good. You're going to hold it at about a 30-degree angle. I'll demonstrate this in class. And we're basically going to push that those cells in that Congo red across the entire slide. So basically, hopefully we see, a nice even smear of cells across the whole slide. Okay, and then we're going to air dry that. So you're probably going to want to prepare these cells first and then start doing your gram stain. Okay, once that's air dry, you're going to flood the entire slide with Manival's stain. And this is a basic dye. This staining procedure actually still works pretty well even if you forget to add the Manivals. But because the Manivals is a basic dye, it will stain the cell wall a little bit. It will penetrate through the capsule to some extent. And then, so the background is going to be stained by the Congo red, and then the mandible stains the cell wall a little bit. So then you see this nice kind of halo effect, which is the capsule if it's there. Okay. So air drying it, this may take maybe 10 or 15 minutes. It kind of depends. As long as most of it is air dry, then you're in good shape. The mandible stain, whatever your book says, I think it's 1 to 2 minutes, something like that. And then you're going to really gently rinse with water. That mandible stain is also a pH indicator. So depending on how it reacts with the Congo red, we get really kind of a lot of different background colors. It might be gray or blue or red. It doesn't really matter. Either way, it's going to give good contrast. So again, after the 1 to 2 minutes there, you're going to gently rinse that off. And then, of course, we always blot it dry before we take a look at it on the microscope. So what are we expecting it to look like? Here's a really nice capsule stain using our method. We used to use Klebsiella pneumonia. Remember, we're going to use oxytoca. Looks the same under the microscope. A good capsule stain is going to have a nice even background. Again, it doesn't matter what color it is, so make sure you look at other people's slides. We see a lot of variation with this one. Sometimes it's red, sometimes it's blue or gray. You will probably see a lot of cells that look like this. So again, usually that mandibles penetrates a little bit. We see kind of a nice halo effect for that capsule. Okay, notice how short and kind of chunky these cells look. These are rod-shaped, bacilli-shaped cells. This micrograph right here, this is the same magnification, I know because I took this picture myself, as this picture right here. It's the same organism. Okay, so notice how much bigger the cells look when we capsule stain them as when they're gram stained. Okay, remember the capsule is not visible because we've heat fixed these cells. So that just tells you how big and puffy the capsule is that these cells are making. Okay, so that's just a nice comparison there. Okay, thanks for listening.